Okay, so we are recording the webinar today. If you registered and are on this now, you will receive the recording afterwards and a link to the slides normally. So yeah, please feel free to introduce yourselves. I can see we've got people where it's evening time in the chat. So welcome, whatever time it is where you are today. I'll be doing an intro to protocols.io, some background context, and a bit of a demo. Hopefully my internet will come along with me today. A little bit of housekeeping. So as you just saw, I've started a recording and you'll get a link. Closed captioning should be enabled. So if that is useful for you, you can use the closed captioning. There'll be time for Q&A at the end. You can pop questions into the Q&A, ideally in the Q&A rather than chat, so I can just look in one place. You can pop them in through the webinar. If I spot them, I'll answer them if I see them in time. If I don't, I'll have a look at the end. There's a couple of other links here. We do these rolling webinars. This one is very much an introduction, but there are more advanced ones and tips and tricks, so you can have a look there. And if you're starting to use protocols.io and would like a more tailored help session on a personal demo, you can also follow that link to sign up for a demo one-to-one, or one of us with a couple of you if you're working together using protocols.io. So feel free to use those resources if it's helpful to you. Quick look at the agenda for today. So yes, I'll do an introduction. I'll just give a framing of why method sharing matters. So protocols.io obviously is a platform for developing and sharing your research methods, but perhaps people haven't realized the significance and why it's valuable to actually do that and be consistent about sharing the comprehensive details of your methods. Kind of introduce the concept of modular publishing and what that means in the context of your research. And then we'll do a demo, as I said, on the platform. I'll show you some protocols. I'll show you some of the functionality that we have, and then hopefully time for questions at the end. So protocols.io, sorry, let me just do something so I can keep a bit of a track, start the timer so I can see a little bit how long I'm talking for. It was a small startup founded in 2012 in California, Berkeley, by my boss, Lenny Tatelman. Some may have heard of him, with some colleagues, and was very much startup for a long time until we were acquired by Springer Nature in 2023. So I joined the team in 2020. Before that was a journal editor, before that a researcher too. So I have been very much head in thinking about research, sharing research as a whole in terms of papers, and then now here thinking about sharing the methods that you're using. So a little bit there on the platform usage. Has almost 200,000 users that we see regularly using the platform. Public protocols, well over 23,000. This was updated earlier in June. Actually, I think I updated it more recently than that. But anyway, fairly recently. And then private protocols, many, many more private protocols than public. So it has two sides to it. The public side are these openly available, open access protocols that anyone can come along and have a look at. I'll show you a bit how you can search for something there. And then privately is if you're using the platform to develop, optimize, create different versions, try different things. You can keep that content private on the platform if you want to do so. The private side is more paid. The public, anyone can use, create a protocol and publish it. Absolutely anyone can do that for free. If you want to use that private side, you will need, or ideally you might have a paid account. The main business case here is that universities, institutes take licenses. So I'm just showing here, we have a lot of academic customers, and also some government and corporate customers as well. So many, many users globally on protocols.io. And our mission's really simple. We want to make it easy for researchers to share their method details before, during, and after publication. So as mentioned, that private side allows you to be developing, can work collaboratively. So very much like a Google document or a document that's shared, multiple people can be in there editing, making changes to a protocol as they go along, until you get it perfect. And then through the publication process, if you're writing a paper, you can link to the methods on protocols.io very easily, make sure that they are publicly available under CC by license, and link to from research papers. I'll give an example of that in a moment. So as I said just now, the two sides, I think this is the slide I updated. It's over 24,000 public protocols on this slide. I clearly just didn't update the earlier one. That open access repository, everything there that's publicly available is under the CC BY license, which is essentially just a fully open access available for reuse license. And can be accessed by anyone globally anywhere. You don't need an account either to access them. You should just be able to access them. If you want to interact, you would need to make an account. So if you want to comment or something like that, you need to make an account. And then the private side, private collaboration tool, where you can securely, everything starts out initially privately, and you choose who to share it with, when to share it, if you want to publish it. It's all fully under your control. I did see a question about limitations of file sizes to create protocols from PDF files by ChatGPT. Well, so first of all, it's not ChatGPT. I'll show you that functionality in a little bit. I don't think there's a limitation to the file size, although there is a limitation to the amount of content you can have in the drive space. But I might not be able to properly answer that, but I'll check it afterwards. But I'll show that functionality in a bit that I can see is being asked about. Okay, so as I mentioned, there's a paid version, and some of your organizations may already have access. So if you did want to check if your organization has got that premium protocols.io access, then if you navigate to this page here, the plans / academia, partway down the page, there's a drop down to check if your organization has premium access. And all that this means is that you would be able to have unlimited private files. You can see on the left up here, the open research plan is available to anyone for free, but you are limited to only 2 private protocols in that instance. If you have 2 private and you publish one, then you have another private. So you can iteratively still use the platform that way, but it has some limitations in terms of the private content. But it might be worth checking if your institution has access. And so why do we care then about the methods? Well, you may have been aware that many people have commented publicly about difficulties of reproducibility in research and issues that arise due to research not being replicable, essentially. And it's true that when you look at research papers, it's very, very hard to find sufficient information about what has been done in that paper for you to be able to reproduce and run the methods and realize or really understand how they generated the data that they generated. So we have a thread of many tweets from X. We'll be working to replace this over time with other sources, but this is what we have historically from people in different disciplines. So there's biologists on the left here, currently reading through so many papers to better understand, but you don't get sufficient information as described elsewhere. It's very common to see that written in a paper in the methods section. And so this person was appreciating methods being shared on protocols.io. In the middle there is physicists who have gone on the chase back through old papers, looking for information about how the devices were fabricated, and you get as previously described, get all the way back to the paper in 2009, devices were fabricated with conventional methods. So we are none the wiser at that point in time. And then, yeah, you can just see in that last tweet the same thing, you know, as described in, nobody actually knows, but they all just say as described in, we've vaguely got something to work. I can see another quick question. Can I confirm that methods that remain private are not novelty destroying with regards to patent application? If they're private, then no one else will have access to them. You completely control who has access to them. So while you keep them private, that would not have any issue for patent, but you would be able to give private access to within a patent application if you wanted to do so. I can kind of show you how you can do some of those things in a bit. Okay, so back to this kind of issue about, you know, the problem of whether methods are available or not. There was a project a little while ago now, this one, the Cancer Reproducibility Project. The authors at Centre for Open Science and Science Exchange, they undertook an initiative to try and reproduce the experiments from 50 published perceived high impact papers that had been published around cancer. And the quick answer to this issue of whether you could reproduce those studies is that no, you couldn't. So the authors contacted, or the people trying to reproduce the experiments contacted the authors of those papers, looked in the papers and asked the authors, can you give us the information about how you performed those experiments? And they could not, is the answer, the very quick end to this scenario. And so the initiative was halted after they just tried, I think, only 18 of the 50 papers because it was too expensive time and money. And in a more recent write-up about this on the Centre for Open Science website, they actually note that 0% of the protocols were completely described. So we can't get the information, can't get it in the papers. Actually, we can't get it from the authors because even their internal mechanism to document what they've done was very poor. So they didn't have sufficient information either, which is terrible. If you think about if they wanted to reproduce, they probably wouldn't be able to replicate their own work down the line. And that was 2017, but this is 2025. This is a very recent piece that was flagged up in a Nature News article about the study. I think it's only been published as a preprint just now, but there's a huge reproducibility project in Brazil that has been looking at common methods rather than in a single field, and again has really illustrated that this remains a big issue. So we're not having enough information about the methods that people are using across many different disciplines, biomedical studies, to be able to reproduce the research. So it's just kind of creates a bit of a question mark. So I would ask you all to think a bit about modular publishing. So the mainly recognized currency of success to assess researchers remains the research paper at the moment. However, a lot of work is being done to try and overhaul the research assessment procedures, processes in different countries and, just everywhere, essentially different institutions and globally, so that researchers are not only assessed on the paper, because there are all these other research outputs. This brings in this modular research output kind of consideration that there is data, there's the methods, there's microscopy images, there might be presentations or posters or other syntheses, molecular data, code, etc. All of these different items can be perceived as important research outputs individually as well. And so if you think about publishing a paper as you go forward, this is a good example on the right here of a paper in Nature Communications from last year, where they have expression constructs deposited at AdGene. They have 2 different protocols on protocols.io, and they have mention of where data homology search is here and the structural data protein data bank. So we have all these modules of the outputs that are being created by these researchers, and they're all noted in the data availability statement. Doesn't always have to be that way. The pointers to methods can happen in materials and methods, but this is I think this is what we need to move towards, to have more reproducible studies that can be reused and then are more valuable over time, essentially. Important also to note that when you divide up into those various outputs, you can get much better recognition to the people who've actually done distinct pieces of the work. So this is that same paper, and you can see Elizabeth Bell is first author, and if we drop down the author list, Kelsey Ramirez was the 4th author on the paper. If we look at the 2 different protocols that are on protocols.io linked to in that paper, Kelsey Ramirez was the 1st author of both of those. So what's really nice is that Kelsey can receive recognition for the development of the methods. Often, I mean, a PhD student tenure of a few years may be that it's spent solely optimizing a method without sufficient time to then apply the method, generate the novel results maybe that would come from that application, but a huge amount of work may have gone into developing the methods themselves. And, this Kelsey is clearly, the expert in the mass spec that's being done here and is the person who would be the expert and deserves recognition. This is the person you should go to if you have questions about the method. So we allow for much better and clearer recognition to the correct individuals by dividing up the And it's very interesting to look at the metrics a little bit. Sometimes on protocols.io, we have every protocol has a metrics tab. So we can see how many views have happened of the protocol across all the versions and various other metrics too. People have run different comments and things that I'll show again in a moment. And we can compare these actually to the accesses of the paper. And this wouldn't always hold true, but this is a different Nature Communications paper where the paper itself has had 37,000 accesses, but the methods has had well over 127,000. I mean, this is a bit outdated now, but the numbers would still be very separate and distinct. So it's clear sometimes that the method might be more of interest, the details of the method, than the paper depending on how you would be reusing that information. But it's nice to know that with the method as a separate output, you can actually track the use, the utility, the reuse of the method this way. So these are protocols for modern science. So rather than having a narrative few sentences in a paragraph for materials and methods of a paper, you can have a method on protocols.io which allows you to capture very clear step-by-step instructions on how to perform an experiment. You can divide up into sections as you see here, and the sections create a table of contents. Though this becomes a method that you can use because you want to reproduce, you can actually run the protocol here from protocols.io. So there's a lot of additional functionality in here that is just leaps and bounds beyond what's possible from a research paper. Commenting is possible. I'll show this again in a moment on the actual platform. You can create copies, forks and versions, so you can adapt a protocol. Create a fork of it means make your own private version of someone else's protocol that you can then adapt. So perhaps you want to do cut and run in a slightly different scenario, or you have slightly different equipment. You can make a fork of this, make the changes for how you would run it in your lab, and do things like this. Sharing, publishing, get DOI, you can submit for peer review. So the protocol's going to be linked into papers, and some journals very easily will oversee peer review of the paper with the method in it on protocols.io as well. In a way that you can't do in a paper, you can also add images, videos of how to do something, photos of a setup, all those kinds of extra pieces of information that would help with anyone that wants to actually run that protocol. And it may be people in your own team that want to run the protocol. So maybe it's for training new and onboarding new students or group members, postdocs who join the lab. This is our standard protocol for doing something. If you have it on protocols.io, can make that a lot easier. And this is how it looks if you drop it into a paper. So on the left is protocol, actually a collection of protocols on protocols.io, which has the DOI. And it's linked with PLOS biology paper. So we can have a nice bidirectional link. So we can link to the PLOS biology paper from the protocol. And if you go to the PLOS biology paper, in the materials and methods section, it actually says methods and protocols for these various different experiments are available as a collection on protocols.io and has the DOI there. So this will link back to the collection of protocols on protocols.io. And in something that we call dynamic permanence, it is possible for an author to update in a new version a protocol on protocols.io. The DOI in the paper will always take you to the version that was available at the time the paper was published. But if the authors have subsequently updated or made new versions available of the protocol or protocols in a collection, you would always be alerted to the fact that there's a newer version when you land on the version from protocols.io and you click across. So you can be aware that the author has made changes and updated that collection. Publishing the protocols on protocols.io is a very, very easy process. Maybe this speaks a bit to the question as well about content remaining private and not losing the novelty, because you have full control over when anyone can access anything. If you want to publish, there's a very obvious green button, and there is a 6-step process. We will prompt you to fill in some pieces of information. So we will require a brief abstract, and we will require authors, and we make you think about whether you should be mentioning funders and things like this. And then if you have a premium account, it will go live immediately. If you don't have a premium account, it will enter a screening process. So we just make sure there's not nonsense going onto the platform. So we do a very rudimentary, very like a preprint assessment of it being something that looks reasonable for us to post. So you can do that and it will go live. If you click reserve DOI, it's a very similar process. At the end, you will have a DOI that won't be live yet. So you could put that link into a paper, but it will not resolve until you come back and fully publish it, or if we find it in a published article. But you'll also get a private link. So editors and reviewers can click and see the private protocol. It's still not publicly available, so only someone with that private link would be able to access it. So it's very, very controlled. Other options here too, as I mentioned, we have some ties with some journals. So you can click to see peer review options, some journals that have very specific partnerships with us to oversee review of a protocol as part of a research paper. And dividing out the protocols this way into different research outputs, so you have paper and protocol, can really help with discoverability and reuse. So this is a nice example we like to show, where at the top was a tweet where someone was looking for RNA extraction method from, and they're working on primary cortical neurons cultures, so some kind of brain cells essentially. And on Twitter, this was someone in Chile, I think, posting in Chile, and then a postdoc at UCSF actually said, oh, by the way, if you're looking for this, there's a method for RNA extraction on protocols.io. And sure enough, so this is the protocol on protocols.io. It was actually linked to from a paper that was published in GigaScience. And if we go to the GigaScience paper, it was a methodological approach and it has that information and links to the protocol, but the application in that paper was on a 3-spine stickleback parasite, so a fish parasite, which if you had not had the protocol as a separate entry, a separate findable, distinct, discoverable output, it's very unlikely you would have found this method if you were looking for your RNA extraction protocol, that you would have thought that this paper looking at a fish parasite would be a good place to go to. So what we have is this distinct output, which clearly has a purpose as an RNA extraction protocol and can be applied in many different scenarios. So we pull it out from the application and it makes it much more discoverable and reusable, and allows for these connections between researchers who are focused on the methods and the techniques rather than the application. So if you do use protocols.io, there's a few different ways you'll interact. So we have a workspace. So workspaces can be public or private. If you look on the platform, you will find some public ones, some you might be able to join. If you create one, by default, it will be private. You can choose to make it visible if you want to. So if you wanted a lab one for your whole lab with, as protocols are made publicly available, they appear in this workspace, then you might want to have it public. If you're all just going to be working behind the scenes, then you might want to keep it private. When you're in the tool, you'll be working in a file manager, which will look very familiar to everybody. It's a typical file manager structure. You can create folders and organize content in there. And then there's the editor view, which is our tailored editor to create that step-by-step format that we have. Okay, so now I'm going to do a bit of a live demo. So I'll just stop the share and then I'll have to reshare. So give me just a second as I combat all the windows on my machine. Okay. So now I've got quite a few tabs already loaded. And this is, if you come to protocols.io/welcome, that's the kind of first page that you would come to. Then here you can just play around. We have quite a nice sort of video demo, which is, I won't let that run actually because it's going to be using up my internet, which just runs through and it has a nice overview of the concepts of how protocols.io works, which is quite nice. But from here to, I'm signed in, so you can see I'm signed in, but if you weren't signed in, this is the place where you can create an account and sign in. But you can also just do searches here. So if we just did a very basic search for PCR, Hopefully that will work for me. That's right. That will bring me up protocols that are publicly available that have the word PCR somewhere. So you can see it's matching in where it's matching in different things that are available. And so any of these you could open, make a fork of, play around. You can sort by different things. How many people have said it works for them? There's clearly one that a few people have said, and this one has high visibility. So you can look around for things that clearly have been working well for other users by searching here. You can also search for people, materials, workspaces. So maybe you might want to look for a workspace on organoids. You can do that here. So you can find, maybe there is a really nice open one, organoid and assembloid workspace that would be open for people to join. So you can find workspaces that might be relevant to your research, where if you join them, you will be able to engage with the other members in that workspace. You can start discussions. If you joined this one, I'm not in it on this account. You can see the protocols that have been published in here and so on. You get the idea. So that's if you want to search for content on there. This is that protocol I showed the screenshot of earlier. So this is a published protocol. You see it has its DOI. You see it's linked to an eLife paper. So in that eLife paper, they have a call out to this protocol. So I think very important to note that although we were acquired by Springer Nature, as this modular tool that creates the output of just the research method, it can slot in to any publication with any publisher. And we see support from all publishers essentially. So if you look around on other journal websites, different publishers, in the instructions to authors, you will often see recommendation to use protocols.io. So you can have the abstract, before start information relevant that needs to be done before you actually start the protocol, and then all of the step-by-step methods that you have. There's that table of contents I mentioned, and that allows you to navigate as well. So you can, I know maybe this phenol chloroform extraction is day 2. So I'm going to jump here on day 2. So you can see where you're getting to. Can use these components to add a timer, temperatures, quantities, and if you add this kind of information, then it can be run when you run the protocol, or you can scale amounts when you run a protocol. So if you're making a buffer or you're making a master mix, you have all the amounts, and one day you want to make more just to create stocks or something, you can scale up or scale down the protocol automatically. I mentioned commenting very briefly earlier. You can see here, there are notes on the right-hand side. These are where other users of protocols.io have had a question about this protocol. So they have wanted to ask something specific to the authors, and the authors do reply. So this is an extreme example. It has many, many comments and answers, but that's because these authors generate new methods. So people are coming and trying to run their methods. So it's very typical that these have a lot of interaction, but you can see it's very constructive questions and answers, trying to help people who have questions, trying to rerun these methods. And if we jump back to the top, I did mention briefly earlier, works for me when I was looking at the search, there's a thumbs up button. If you click it, it adds a number, someone else to the people who have said that they can actually get this protocol to work for them. So it's not just that I like it, it's actually how it's working. And if you click, you can see all the people who have done that, and they're not all just members of Stephen Henikoff's group. It's many people in many different places who are managing to use these protocols. Lots of other places to add guideline information warnings, you can add ethics statement, etc. If people make forks and changes to the protocol in their own versions, you can track that here. Mentioned the metrics earlier. You can see if the protocol has been cited somewhere, you can actually see, again, this is an extreme example, but this one has been highly cited because we know it's very, very highly used. It's got almost 250,000 views of this protocol across all versions. I mentioned making new versions earlier. If you do make new versions and you publish them, you'll always be able to track back through the earlier versions and see the dates of them. So it's very easy to go back, and if you want, you can do a comparison to see what was updated from one version to the next. So you'll be able to see highlighted where the changes were made between versions. So that can be really useful Similarly, if someone has made a fork, you can see the changes from the original to the forked version of that protocol. So lots of nice functionality on there. Was there anything else I wanted to say? Well, when you're looking at the actual protocol itself, it's very easy to do the fork. If you want to do that, you can click here and it will walk you through making a fork where you want to put that. So you can select the folder where you will create it in your workspace and it will make you your own private version of this. I won't do that just now. Copy, only you can make a copy. So if you've made your own protocol and then you've got another one that's slightly different, you don't want it to be a fork, you want it to be a distinct one, you can just make a copy. So you can also create a template with all your regular metadata in it if you want, and then just use that to create a copy each time. If you want to run a protocol, you can start a run record. So you can click run, maybe go to your run records folder and start the run. I'll just do very quickly and show you, although I won't get into too much detail, but the run allows you to walk through every step and say when you've done it. I mentioned earlier, if you've put a timer, you can actually run the timer during the run. I'm not going to let that run for 3 hours or 3 minutes, whatever it is, 3 minutes. But I can say done. Here is where there's a quantity. As I mentioned, we can scale if we want to. So if we come over here, we can scale up. So if this was something that wasn't just a single item, but a recipe for a buffer or something, I'd be able to scale the whole thing, save that, and I can see the changes that I've made. Can also add comments, so I can make observations during the run of the protocol and keep my own observations, make comments, things that I did differently, something that I'm trying differently, or something quirky about the setup that day, anything like that I can run through. And this will be being saved all the way through. I won't finish the run, but if I go back to my file manager and click into that run records, you'll see it started making this run record today. So we're 21st of July and I'm in the UK, so it's 11:34. And it's grey, it's incomplete. So if overnight I've kind of got partway through and I need to come back to this tomorrow, I can close everything down, it will all be saved. And tomorrow I can come in and click resume and pick it back up again and just start again. If I'm in this workspace and colleagues are in here too, then my colleague could finish this. If for some reason I wasn't able to come back to it tomorrow, they would be able to pick it up and finish it as well. So there's a lot of nice functionality with the run records. But that brings me nicely to the file manager where we are here. So this is where you'll do all the work. You can create folders, like I said. You can see there are different colors to the icons. So the orange are the private protocols. The green here, that's a completed run record. So that's a finished one. When it's finished, you can no longer make any edits to it. And you can see that it's fully saved and completed. This is really helpful to correlate what you did on a day with the data that you created that day. So if you took a lot of microscopy images, for example, you can have the run record for how exactly what you did that day when you were preparing the samples, or whatever you did differently, how you actually configured the microscope, whatever the set of information is that it's important to keep with the data. You can have that time and date stamped. This one was performed June 12th. I know that it was me that did it. I can look at anything that happened that day and see the changes that were made. If you belong to many workspaces, they'll show up on the left-hand side here. You can open out, you can see, however would make sense. If you have the license to create more and have more private files, you can have workspaces for different projects or teams. You might have a central umbrellas workspace, which could be for the whole lab, maybe an individual one for your working, things like this. Within the workspaces, you have the person who creates it will initially be the owner and the administrator. You can make other people administrators as well. And there is a lot of different settings, and I'm not going to go too deep down the rabbit hole, but would encourage you to look at what's in here. Most importantly, if you're trying to set one up with colleagues, you can add different members. So if you go to that administration and members, you can here invite other people. You can either do it by email address, or you can have a link. So if you want everyone in the lab to join, you can generate a link and give them the link, and it will invite them to that workspace. Generally, you probably want to control who's joining your workspace rather than have it be open for anyone to join. You can do this all here. Other settings and things like the visibility of the workspace, you can do in the general settings that you have in here. Or delete things, etc., etc. There's a lot in here for and security and privacy. This is the visibility settings. So this is private, but we could make it public. So if you wanted a public lab workspace, you could change that here. And you can control how people are able to join the workspace. Many different things. I won't, as I said, jump too far down that rabbit hole because there's just so much in here. You can go between the folder and a profile view. So if you do have a public one, you can have a profile view, which will allow you to see what's been done. That's a bit like the organoid one. This is that profile view of that organoid one. I'm not a member, but I can see it. So it's about what's visible publicly a little bit when you have it like this. I was then going to show how to import protocols. So there was a question earlier about adding files using the PDFs, the importer process. So if you already have existing protocols in some format or another, so they might be a doc or they might be a PDF, and I was just going to show what those look like actually. So this one here is this PDF. That's that PDF that I have in the workspace. It's a protocol file that, you may already have existing files in your labs for methods that you've been working on. The other one that I have in there is a doc, which is this short RNP into Dino. I will attempt a live demo of the importer. So this is a way that we have created to import protocols that you already have in a different format, because you maybe have them as docs on different laptops, or you maybe have a centralized Google Drive or something like this, but maybe you want to get them onto protocols.io, but don't want to have to write everything out again. This will allow you to do it this way. So when you click new, you can go to import a protocol, and that will prompt you to upload the files. I've already uploaded these files into my workspace. So if I click on one of the files, you can see it says create a new protocol from this file. So very important to note, it's not ChatGPT. There are issues around privacy with ChatGPT. This is using OpenAI GPT-4, and none of the protocol content that's uploaded and we use the importer on, it never gets sent back to them. So they will not have any access to any of your data. So I've clicked that now, and it's going to start making a protocol for me from that PDF in the protocols.io format. So it sometimes takes a little moment. It depends on how long the protocol file is, depends on many things. I've just been out on a week's holiday. I believe they've released a new version, and it's now much more accurate than it was before, but I haven't yet tested it. So we're doing a live first test for me here today. But it can take a few minutes, I think. So maybe we'll just leave that, and I'll come back to it in a moment, and hopefully it will upload. But while that works behind scenes, I'll show you the editor. So this is just a demo protocol that I have in protocols.io. If I started and made a new one, it would be completely blank. And you can see, though, I've started creating the protocol here, just adding some things. So we have some sections, we have some steps. Very easy to make a new section, and the colors come there. So I'm not sure where we got to, but we can say 5 and 6. And with those sections, that's generating the table of contents for me and the color coding that you can see that's coming with. We'll close that again, pull that out. We can add new steps as well, and just keep adding things that we have. You can have sub steps, you can have a step case. So if a protocol has 2 different pathways at a certain point in time, maybe one at 80 degrees, one at 50 degrees, or whatever the different categories might be, you can have 2 different pathways to follow at a certain point in time. You can't merge them back, so you would then need to see protocols through to completion. But in the steps, here we have the components. So like I mentioned earlier, you can add quantities, or timers. You can add reagent information. We pull in information from a lot of catalogs, vendor catalogs. So we, you know, down to the catalog number, can have information about the reagents that you're using. Or you can define your own reagents, you know, A and B here. And the components are all on the right-hand side. So I'm just giving you a glimpse. But if you want to do an amount of something, you can see you can just add here 100, and you can choose what the units are that are appropriate in that instance. You could put the reagent info here, or you could just have it like this, and that gives you this formatting that you saw in the run that allows you to scale and things like this. You can add in tables. You can see we've got the table at the top, which is very familiar format. If you put this in and actually add in Excel formulas, then if you complete a table when you're doing a run, it will perform the equation that you've put in. So it's not a replacement for Excel, but it does allow you to do some rudimentary calculations on the fly if you have preloaded that information into the table when you make the protocol. Smart component allows you to kind of do a key value pair, we call it. So you can have a question or something that you need to be looking at as you do a run of a protocol, like number of cells or percent conversion, something like this. And you can require an answer. So if you're doing a run, you will have to complete that value before you'll be able to say, I've done this part of it. And you can export out the inputs from those at the end of the run as well. So you can download as a CSV. So all sorts of other things. I mentioned you can put in videos. You can put in directly if you've taken photos on your phone or small videos on your phone. If it's a much larger video, you can grab embed code from somewhere like YouTube or Vimeo and pop that in. So literally for adding this video here, I'll show you in a separate step. You are just adding embed media and pasting in the code for the video, and it will pull it in for you. So it's incredibly easy to do that. Lots of other things in here. Well plate maps if you're doing screening. You can embed sub protocols. So if you've got a buffer recipe or something that needs to be done each time in multiple different scenarios, you can make a small protocol and then point to that protocol within. So you can have a sub protocol. You can only nest one layer because it becomes too complicated to run otherwise, but you can see a lot of the options and things that are in here. Way more than I can go into now. Let me go back and see. Oh, we're still spinning on that. That's annoying. Hopefully it will run by the end because it would show you nicely that you can get in the protocols. I wonder if I can do the smaller one and it might be faster. It may not be liking the fact my internet is not great. This one might be a bit faster if I do this one. Let's try this one instead, which is just that smaller Google Doc one that I had there. If you are short on time and you don't want to wait for these to complete, which you may very well not want to, we also have a service to enter protocols for you. And if you're somewhere with a license, this is included as a part of the license. So with this functionality, you can send us a protocol. We will enter it into the platform for you. We have a team of editors who will do that with research backgrounds. They'll know kind of how something should look, and they will add all the component functionality and all of that extra whistles and bells. If you use the importer, if any of them ever work, it doesn't add the components and things like that. They're not getting added for you. So the import process is a way of making sure that all the functionality of protocols IO is actually applied to the protocols that are being added. And that's very easy to just drag and drop files here, upload file, and put it in. If you're somewhere with a license, it should happen very quickly here. Sometimes some of the journals that we have partnerships with have a promo code to oversee that as well. Otherwise, it's a paid service. But we will normally check with you to make sure if you have got a code or something to allow you to use that for free before we go ahead. That is most of the things I think I was going to go through. One other thing I was going to show, if you're creating a protocol, this is the editor, you can go backwards and forwards very easily to the view and edit modes. In the view mode is where you can see all of those options more clearly for publish, reserve, DOI. The peer review drop down is all the different journals that we have partnerships with. These will not necessarily take you to submission page for those journals, but they will take you probably to a page will explain the nature of the partnership. The most bedded in one that we have is probably PLOS One Lab Protocols, where they require that the method be on protocols.io before, as a part of the paper, very core part of the paper, and so they're bidirectionally linked across. And I was also going to show that you can share. So you can see the share button in here if you're in the file manager too. So if you're not ready to make it public, you can actually just share this with individual colleagues. So you can add someone by their email. So Lenny is my boss. Can find him here. Oh, he was there and then he disappeared. Normally that would work. I don't know why that's not working. He was there. Let's just try Lenny. Yeah, there we go, Lenny. So we can share with somebody, we can change the permissions that they have. If they can view, just preview, can edit and publish, or be a co-admin. You would have to click share as well. You can send them a message at the same time. So that's individuals, you can share this way. You can also share with a workspace. So if you're working in your workspace and you want to share with the whole lab, you could find your lab workspace and share with the whole lab that way as well. So lots of different ways of sharing privately this way if you want to do so. Oh, he is on there. You can also get a reviewer link. So if you just want to get a private link to give to whomever you want to have access, so either for reviewers and editors at a journal, you can click get link. And oh, I don't actually own this one. That's weird. I'm in a different account to the one that owns that protocol, so I could get the link if it was my protocol, but it's not. Weirdly, that's a quirk of how I've set this up today. But you would just get a link, and then you could put that into the paper, or into a patent application. When I'm thinking back to the question that came up earlier, so that's most of what I wanted to share. Let's see if either of them actually worked. Oh yeah. Yes, we do have them in. So they've created protocols. So in there, that's the RNP. Oh, this is the long one that was from the PDF. Oh no, this is the RNP into Dino. That's the RNP into Dino. So that's the 16-step one that was this here. And quite nicely, you can see it's got my authors have been pulled in, the affiliation. It's put me as the owner because I've made it on my account here. But we've got the abstract before you start on all the steps. So that one was completed. And this one is the other one, which was much more complicated, the big PDF. Hopefully it's worked. Protocol, oh no, we're in the same one. I'm viewing the same one twice. 1 second while I reload. Oh, maybe I clicked the same one twice. I'm not sure. Maybe the other one didn't finish and I'm looking at the same. I don't know what I did. I thought that one did too, but maybe it's only done one. I've confused things, perhaps doing them at the same time. But anyway, you get the idea that this one has been created. I've got too many tabs open now. So that's quite nice to show you how you can get from a file that you've already got onto protocols.io. But as I mentioned, you won't get any of the components, but you can further edit this to add all of the other things that you want to. Conscious of the time, so maybe I'll skip and look at some of the other questions before I finish up today. So I'm just going to pull over. So in the case of low dose or low dose rate effects, the reproducibility has been difficult due to missing semantic interoperability. Are there any remedies? I can't answer about specific reproducibility of individual protocols, unfortunately, but this is the perfect scenario where if you find a protocol on the site and you're struggling to reproduce that, you can ask questions to the authors of the protocols themselves. So I think hopefully that would illustrate the value of the questions, the ability to ask questions of the authors on there and directly interact with them there. Next one from Brian, should I make the protocol public after submission to a journal, or is there a way to keep it private but have a link for reviewers if they wish? Yes, so I think I hopefully have answered that. You can do the reserve DOI, which will give you the DOI to put in the paper, but also a private link that you can give to reviewers and editors if they want to look at things. And your second part of your question was, do any journals have issue with the protocol being publicly available on protocols.io beforehand? We've never come across that, but if an author has any hesitation, they can do that reserve. So you can, I mean, we would always suggest making it fully publicly available. Earlier the better, just because sharing helps science go faster or research go faster. But if you're not comfortable, you can do the reserve DOI, so that it's not public and you can coordinate the paper and protocol being published at the same time. So that's done. Are there any good tools for comparisons of different protocols produced by ChatGPT? I really don't know the answer to that. And I mean, I'm not sure if people are using ChatGPT to create protocols, and probably the best way to test if they work is to just try and actually perform them. So it's a bit hard to answer that question, I'm afraid. I'm not really sure I can answer that one. In the free version, you mentioned only 1 or 2 private protocols. Was this per user, workspace, or org? It's per user, but it's a bit complicated in that if you create a workspace and add in your colleagues, that workspace will be limited to 2 free private files. So you do have to be careful. It does have limitations, you just have to play around with it. There are some ways, if you kind of invite a friend, sometimes you can get an extra free private protocol. So yeah, it's limited, but you can use it, the free version. It should be workable. Next question was, I saw integration with other data management systems. Is the API free to use? Currently, yes, it is. And we do have several integrations. As you saw, we've got integrations with external data management systems, as you've seen, and also electronic lab notebooks, some. So you can, if you have people there who can build up an integration, you could create your own integration into protocols.io. We sometimes build integrations if we know there's a need, for example, with specific data locations or ELNs if we get requests. So hopefully that answers that for you. Yeah, OpenAI, that's what we're using. She had asked that question. We're using OpenAI for the importer. And as I said before, we do not send any information to them for learning. So nothing that you put into our importer would be shared beyond. It remains private and will not be shared on. So that is probably important to clarify. Then you asked, data integrity needs to be maintained by each user team. Yes, but it will, as long as you don't share, then everything is very secure. We have had to jump through a lot of hoops to make sure that everything is very secure, because we work with some pharmaceutical companies and we work with some government agencies, as you can imagine. Actually, some university infrastructure that we have interacted with have been very demanding in the security requirements. And we're obviously compliant with things like GDPR within Europe and other equivalents in different countries to maintain data integrity and user integrity. But within your workspace, you obviously control how you share information with other team members and things like this. Quite a few questions today, which is great. Am I aware of other companies doing what we do? Open Wetware comes to mind, but it's down since April. Is the value submitting to protocols.io and another site, at least hypothetically? So yes, Open Wetware was probably the best example, and they are no longer functioning, so that's not around anymore. Some electronic lab notebooks have protocols parts, but none that allow you to publish the same way that we do, and none that are as tailored to step-by-step methods as presented on protocols.io. So for the time being, I think we don't have big competitors. There are a couple of new emerging. There's something called Potato, and I haven't really looked at it recently. There was something called Briefly Bio, but they're very much, I think, still startup phase. So I'm not really sure what the current status of those is. As to whether there's value to submitting to protocols.io and another site, at least hypothetically. If you want to get a published version with a DOI, then I would say protocols.io is your best bet. If you've published a protocol in a paper somewhere and it's in a kind of PDF format as a supporting information file, I still think there's value to adding to protocols.io, because then you have this dynamic interactive version that you can make available that other people can build upon. If it's in a PDF, it's much harder to find. You can't fork and kind of run it very easily. You can't adapt it for yourself without essentially rewriting the whole thing. So I obviously have a conflict and a bit of a bias, but I would say yes, there's value to doing that. Hopefully that answers the question. Jan is asking, if I start reproducing a protocol, will its author be notified? No, they can configure to be notified if someone performs a run of their protocol. But if you make a fork of their protocol, a private fork, they will be able to see how many private forks exist out there, but they will not know who has created a private fork. And then if you perform runs of your private version of their protocol, they will not be aware of that. You may want to correspond with them if you are using their protocol or doing things. If you made the fork and made many changes and then ultimately published it, they would see a public fork. So you can track if someone makes a fork, eventually makes it publicly available, that's visible. You can look under the forks tab of a protocol to see what's happened there. If you're reproducing it and you publish a paper having reproduced their protocol, obviously we might hope that you would cite either their protocol or the paper that it came from, or both, if that was appropriate. So hopefully that answers that question. Maged asks, can I publish a protocol for engineering design process? It's really versatile tool. So although originally it was developed with a view to kind of being something that worked for wet lab research, biological life sciences, these days it's incredibly versatile. So we do see things like device construction for engineering things, or tools, microscope setup, just complicated setups, computational workflow pipelines, anything that has a step-by-step process you can put onto protocols.io, because it has got this very, very versatile structure to it. So I think it's, ultimately can be used for anything, really. So would definitely recommend that you play around with it, really, and see what you can do. I'm conscious now, I've just remembered I did have some other, I think that was all the questions that were showing up. I don't think there was anything in chat. I did have a few more slides, but I think I've probably gone through most of the important things. It was just, well, actually one quick thing that I'll show. I realize we're almost at the hour too, but let me quickly just show this. Beyond wet lab methods, that was just to illustrate that there are a lot of other, well, are you seeing all the Zoom chat things too, or are you just seeing the slides? I'm not sure what you're seeing now. I've got everything's come up on my screen. But anyway, you can see there's many different kinds of protocols that can be shared. I can share these slides afterwards as well. And there were some nice quotes from people about how they've used protocols.io and how it's enhanced their career. Won't go too deeply. Oh, and mentioning that many publishers and journals have also recommended, beyond Springer Nature, obviously, funders too, and institutions. And then there's lots of links to all the different help resources and tools that we have on the platform. So you'll have the links to all the slides on there afterwards that you can play around with. And that webinar's gone. So then if anyone has questions afterwards, you can feel free to drop me an email, emma@protocols.io, or our general one is info@protocols.io. And before people go, I can see there was one more question. I'll come to that in just a second. I'm just going to pop a link into the chat. We have quick surveys for the webinar feedback. Obviously that slide is up there, the QR code will work for you, but the link for that, if people can just take a little moment to give feedback, can help us to adjust the webinar, improve the webinar, change the content, or think about other things that it might be good for us to present another time. So I've popped that link in there. And the last question I think I see in chat: how much of a modification on an existing protocol becomes a new protocol? Yeah, difficult for a general answer, but who might oversee this? That's a kind of how long is a piece of string question. But I think if you've made enough of an adaptation that you think it's still worth sharing a new version of a protocol, then it's probably worth publishing. You know, if you've adapted and you're publishing a paper because on using an adaptation, but obviously you would give credit, cite the original one as well, credit the original authors, but it might still be valuable to share the adapted version of the protocol too. So, I mean, you have to use common sense, maybe as a courtesy check with the original authors if they have a strong opinion about it, and then go from there, really. Hope that makes sense. I am still around for a couple more minutes. If anyone has any more questions, feel free to drop them in. But I realise we have gone slightly over the hour. So if people need to go, then thank you very much for joining today. I hope it's been useful. Please do give feedback so we can continue to improve. Thanks very much, everyone. Oh, I'll stop the recording too.